Kia ora koutou, no mai haere mai. Welcome back to all those who've been joining us today, and if you're just joining us now, welcome. We've got a really exciting afternoon session. It's slightly different to what might be on an extra talk to make up for the lost talk in the last session, which is quite cool. So thank you for joining us slightly earlier than what was originally planned. I'm going to hand straight over to Simon to introduce the next research topic and the speakers that are going to talk alongside it.
Kia ora, Taylor. We're now moving into the research project Conservation and Restoration. It's a huge challenge to conserve and restore kauri and native plants vulnerable to myrtle rust for future generations. It requires a te ao Māori worldview and appropriate governance arrangements over the whenua, not just where adult plants grow, but also where they potentially regenerate. Working from a pathogen host and ecosystem point of view, this research team incorporates conservation biology principles to make sure susceptible plant species survive myrtle rust and kauri dieback in Aotearoa.
Kia ora. I'm Dr. Peter Heenan. I'm a senior scientist at Manaaki Whenua Land Care Research based at Lincoln. I have a background in plant taxonomy and plant systematics and have been involved for most of my scientific career studying native plants in terms of how they're classified and how they're named.
Kia ora, ko Ricky Nelson tōku ingoa. I'm the operations manager for Kauri. We're based up at Taketake. We've got about 3 hapu at Taketake, working as kaitiaki, protecting our sacred taonga in the Ngāhere. My role is operations manager for a hapu-owned entity, which is Kauri, and that is owned by 2 hapu. One is Ngāti Te Wai, my hapu, which I'm the environment rep for, and Taufo ki Otari, who is our other hapu that we're quite closely related to. The kauri, or the kauri and the kaimai, are the southernmost regenerating kauri forests in Aotearoa. So we took a team from Shaun, Manaaki Whenua, up there to collect some seeds back in early 2019. And, you know, due to the observations and so on, they said that this was the healthiest kauri forest in Aotearoa.
So in Rākau Taketake, I'm leading a project on conservation genetics of kauri, and the real idea of that work is to understand the genetic diversity of kauri so we can manage it in response to the kauri dieback. We have very limited knowledge of the natural genetic diversity of kauri across the landscape. So from the far north to the far south, how does it vary naturally? So this project is to develop what we call population genetic markers, which are unique fingerprinting type markers. So if you're watching CSI programs on TV and they're talking about identifying a person who committed a crime and they've got blood or something from the scene, they will take that, and what they're looking for is DNA fingerprinting that can characterise that one particular individual person. And so we're operating a similar way with what we do with kauri. We're looking for, often not at the level of an individual plant, but certainly at the level of population, we're able to retrieve markers that can identify distinct populations. And so one of the things is we want to know if there are populations that have unique DNA fingerprinting that occurs nowhere else, because they would be quite important to conserve.
Kauri, we've been around for a few years. Kauri is now a limited liability company that is owned by 2 hapu. We needed a mechanism to identify resourcing and funding, to bring that funding down, to set up the framework and the structures to employ our people, to train, develop the capacity, capability, and to deliver services so that we can, start achieving some of our aspirations and some of our goals.
So in the case of the Kaimai and the Coromandel area, for example, the kauri of the Coromandel Peninsula might be quite distinct, because to the north, west and south there could be quite different populations, and the Coromandel is known to have some unique species anyway, so the kauri could be genetically different. Now if we come back down to the Kaimais, then we've got a situation where The kaimais are at the very southern limit of Kauri distribution. They're south of what is probably a distinct area in the Coromandel Peninsula, based on what we know about the sort of history of that region and the current flora. And so the Kaimai range is also a bit complicated in that it has deep incised valleys, like a lot of Northland does, and those different catchments may also have a different genotype of Kauri. So in the case of an area like the Kaimais, it might actually be that you need to protect every catchment in there in terms of how you would manage the Kauri. But it's really that understanding that these are our trees and these are how they differ in that unseen way of the genetic diversity that sits in behind what you can see as a giant forest tree in front of you. The word genome is bandied around and it's often a good all-encompassing phrase for the genetics of a plant. You can say it's in the genome. But in actual fact, what we're doing with the fingerprinting that I described earlier is we might work across the entire genome, but we're not sequencing the entire genome. And for something like Kauri, that would be costing multi-millions of dollars because it's a massive genome, and to sequence each individual DNA nucleotide in there would be a massive task. For example, we've just been doing some work on Kānuka, and we sampled the genome, and we ended up with 1,300 what we call single nucleotide polymorphisms. So that just means single means one nucleotide, and one nucleotide that varies, the polymorphism. And we only had 1,300 of those from the entire genome that we were happy with, gave us a rigorous statistical genetic result. And so, you know, that genome is probably, it'll be tens of millions of DNA bases long, and we were only interested in 1,300 that were scattered right across it. In terms of the integrity of the plant, we're not altering its DNA, and we're only really searching for and wanting to understand the variation in these more rapidly evolving parts of the DNA that have no real function in terms of genes. To be perfectly honest, one of the big challenges, as I see it, is that Maori have so much pressure these days in these local, environments where they're deemed to have responsibility for a piece of forest, and they're making decisions about that. They're under-resourced, understaffed. There's often a bit of a scientific gap, and that's quite a challenge as well for the scientists to explain clearly in an acceptable, non-threatening way that we're not going to be sequencing genes and selling those and making lots of money. That's the least of the technology we're using can't do that anyway, but it's a matter of the technology we're using is pretty low impact and it shouldn't be really threatening. There's no commercial use in there. We're using standard scientific methods that are globally accepted. So, but that's a challenge for us to bring to them. And then, as I say, they've got the challenge of their resources primarily.
Back in 2018, 2019, we focused on our rangatahi, bringing them into the kaupapa with the aspiration of developing future employment for them. So we had one of our, she's actually our lead Cody track ambassador, which is Trinity Nikora, and she has been volunteering in this space on behalf of our hapu with the kaupapa since she was 16. She's been employed full-time since late last year with Kaimai Cody. And not only that, she's 19 now, but she's got a leadership position. If you have a look at our team, all young, and there's a reason around that. We needed to make sure that we had a reasonably young team so that all of our youth could look up to and aspire to. We've had a few issues over the generations, unfortunately, with mental health and youth suicide, especially in Katikati. So there's a number of kaupapa that we're trying to drive and support our youth. Our youth need to have hope. And we believe taking them back to our roots, we were actually sea people in the Moana, Taufau, who were the seafaring people, but we also had our kaupapa that in the Ngāhere with the Kori and the rongo and that kind of thing. We know for a fact that when we take our people into the bush, there's a certain wairua in there that heals them.
They've got lots of demands, not only demands for biodiversity, but There's cultural demands of running their enterprise, meeting people's needs, which they take there's a completely different perspective on the world. There's the holistic Maori view, so there's a lot of resources and effort goes into the holistic approach, whereas scientists, we tend to compartmentalise things, and if we go and visit, we want permission just to do kauri from that site. It's a very small part of their world. So, there's a lot of things to understand there, but as I say, the best way to do that is to sit down and talk. And I've got to say, in terms of our hui at Taketake, talking to Carlton for 2 or 3 hours one evening was a brilliant experience. And then on the following morning, standing under a pahutakawa, having a pretty good kōrero with Ricky about the background of that marae and what they'd endured over the last couple of 100 centuries was pretty eye-opening and pretty interesting, and certainly gave me a real deeper perspective of where they were coming from and why Maori have such a broad holistic view and the concerns they have, which we really need to address through Te Tiriti in New Zealand.
We need to recognise the treaty partnership between us and the other partners, which are government entities. And they always talk about the capacity capability building. What does that mean? We need the resourcing to grease the wheel so we can do the mahi. We need to get out there into the field. We need reliable, long-term resourcing and funding so that we can actually be kaitiaki of our natural resources. Collectively as tangata whenua, I think we need to start working together and collaborating. I really do believe that we have some really innovative initiatives down here in Tauranga Moana, where we think outside the square, outside the box, and I think we can, other iwi hapu can learn a lot from us. We absolutely need to learn what they're doing up Northland, what they're doing up at the Waitakares, and as well as Coromandel. We need to be collectively meeting, having hui wānanga, sharing, and that kind of thing. And here's the thing with government agencies, they need to be supporting this kaupapa. They need to be helping to enable us so that we're all working together, kotahitanga, across all kauri lands, rather than just our little plots here and there and that kind of thing, from the northern kauri all the way down to the southern kauri.
I guess that's the spark, is that there's always more stuff to learn about what these plants are, what their whakapapa is, and we look at that through DNA, obviously, and at different levels. So just an understanding how they fit into the environment, and these days that understanding is also the cultural environment, the human impact. If we look around New Zealand, we see large tracts of lowland New Zealand have been cleared and lost, and for productive purposes, for urban settlements. But the impact on biodiversity, and I'm not just talking plants here, it's birds, insects, etc., is massive. We can't keep having the impact we're having without some costs in there. There's got to be a tipping point, and as we hear with climate change and these pandemics that are going around and other pest organisms exploding, there's massive impacts happening. So it's really understanding the biodiversity so that we can manage those impacts and we can look to the future about protecting the resources that we've got left from what we've done to the environment.
Awesome. Thank you to the Conservation and Restoration team for that fantastic video. I'd now like to introduce the next speaker. Emma Simpkins has been the Botanical Records and Conservation Specialist at the Auckland Botanic Gardens for 8 years. She manages the Plant Records database, oversees threatened plant conservation projects, and facilitates research projects based at the gardens or using the plant collections. Welcome, Emma.
Kia ora everyone, thanks for having me. Today I'm going to share just a little bit about our work with Rākau Ngāti Kuri in supporting ex situ as well as some in situ conservation. So botanic gardens globally have 3 key roles. That's in education, research, and conservation. And that fits in that kind of bubble of recreation where people come to a beautiful place and enjoy themselves, but there's other things that go on at a botanic garden, and that allows us to have that clear mandate of supporting conservation in Aotearoa. Several years ago now, there was some DNA work being done on Rātamoiha or Metrosiris bartlettii, and while they were sampling, they realised that the population had significantly declined down to about 12 individuals, which was really alarming. So the material that they'd taken for DNA work also came down to us to propagate. We ended up propagating these from cuttings and grew them on through the kind of tube stage and potted them up. And we now have 5 individuals of the 12 represented in the wild. These plants are planted out in our main garden, and they are mapped so that we can keep a very close eye on which plant is which is obviously really important for whakapapa. We've been working really closely with Ngāti Kuri in terms of their repatriation plan and conservation plan in the far north. And their goal is to repatriate 1,000 individuals back to the wild. So that's a really great, ambitious target and something that we're very excited to support. So over the next 2 to 3 years, every year we plan to grow 500 plants from these 5 individuals and return them back to Ngāti Kuri. Earlier last year, we sent 200 mature plants back to them, which was a great first step in that project. Some of these individuals can be tricky to grow. Who knows what those plants have that are different from each other. So 2 plants out of the 5 do really well at growing in the nursery, and then the other 3 are a little bit tricky, and that's something we'll have to work out over time. One of the great things of being part of a botanic garden is we have lots of networks, and within New Zealand we're really well connected with our other botanic gardens, and Ngāti Kuri and Auckland Botanic Gardens agreed that as well as having this ex situ collection, this permanent duplication of the wild population in Auckland, that because we don't know the ongoing impacts that myrtle rust may have on the wild population, we should kind of hedge our bet and put these plants in other places while we're still learning more about myrtle rust. So we've worked with Otari, Wilton's Bush and Dunedin Botanic Gardens to have material sent down to them as well to grow these 5 individuals. And that's really important to have a backup of a backup sometimes when you're not really sure what the long-term impacts of the biosecurity threats to this plant may be. As well as working on this taonga for Ngāti Kuri, we also share our knowledge and our observations with them. We track their phenology really carefully. We've had the pleasure of hosting one of their people, Thomas, for a summer internship where he learnt about propagating and also about hand pollinating this plant. One of the tricks for Rātamoiha is it doesn't produce its own viable seed, and so we have to do hand pollinations between different genotypes to ensure that we have genetically viable seed that we can grow on in the long term. So that's what we've been working towards, and as those individuals are returned back to Ngāti Kuri, they'll be able to carry on that work as they have been over the last couple of years up in the far north. So kia ora everyone.
Kia ora, thanks very much for that. We'll now move on to a presentation from Karin van der Walt. Karin has more than 20 years experience in threatened plant conservation. In her current role as Conservation and Science Advisor for Otari Native Botanic Garden, she is using the Lyons Otari Plant Conservation Laboratory to establish methods for the conservation of some of New Zealand's rarest plant species. These methods include seed germination, tissue culture, seed banking and long-term storage of seed, pollen and embryo cryopreservation. Welcome, Karin.
Thank you very much. That was a bit of a mouthful, but thank you for the introduction. So today I'm going to tell you a little bit about seed banking and whether that is actually a viable option for our fleshy Myrtaceae species in New Zealand. So before we start doing a deep dive into it, let's just touch base on what seed banking actually is. So seed banking, you would do that so that you have got options to restore species or recover species when they've become critically low, or even restore ecosystem. Many of our food and crop species globally are conserved in seed banks, and that is part of our food security as a human race. Conventional seed banking traditionally means that we dry the seed quite to low moisture contents, 3 to 7% moisture content, and then we store them at minus 20 degrees Celsius. However, seed storage is not a new concept. It's been done in various shapes and forms for hundreds, if not thousands of years. So when we come a little bit closer to home and we start investigating seed banking and our New Zealand Myrtaceae species, we've got 6 native genera in New Zealand, which belongs to the Myrtaceae family. For the purpose of this talk, I've divided them broadly into 2 groups. So the first group is the dry-fruited species, and those are the Kanuka, Manuka, and Rata. And you can see in those photos that came up, at the top are the capsules, and then the 2 bottom photos are the actual seed that comes out of those capsules. And the second group is the group that I'll be focusing on for the next 3.5 minutes, and that is our fleshy fruited species. And these are 2 Lophomerta species, so Rama and Rahutu, and also Neomyrtus, which is also Rahutu, and Maretawake, which is our only Zyzidium species. And as you can see, the photos that once again came up, those are fruit that's got a fleshy or a very moist outer layer with pulp, and then inside the fruit, actually, the seeds looks quite a little bit different. So I investigated how does our 2 fleshy species, Lophomertus and Neomyrtus, respond when we store them at that conventional seed banking, which was minus 18. And what I found is that for the seed that I collected from Rama and Rahutu, did not actually survive conventional seed banking. As I said, that was at 3 to 7% moisture content, and storing them or banking them at minus 20, which is our freezers, deep freezers store at minus 20. For Lophomertus obcordata, they didn't mind when we dried them, so we removed the moisture content, but actually 2 of the 3 seed lots tested started losing a lot of viability after 2 years, and that's not what we want to see, because seed banking we do for the long term. To investigate this a little bit more, I used something called the differential scanning calorimetry, which is DSC. And this is the outcome of one of those graphs. So at the moment, we're looking at seed going from minus 90 to 10 degrees Celsius. So we're reading it from left to right like you normally would. LM in this graph means lipid melt. So that's fat, and the 3 lines represent the 3 species I'm talking about. Now, in itself, these graphs don't look like much, but when we start looking at what's the range of the lipid melt, we can see in Rama or Lophomertus bullata, this happens between minus 39 and minus 16. For Neomyrtus, that's minus 39 to minus 24, and Obcordata minus 38 to minus 23. And this shows, especially for Lophomertus bullata, that their lipids are not stable. They are metastable over those seed banking temperatures. Remember, we store seeds at minus 20. So that range of lipids, they are basically unhappy at that storage temperature. And that's why we're seeing all the viability loss for Rama and the other 2 species as well. The last species I'm quickly going to talk about is our final fleshy fruited species, and that's Maretawake or Zyzidium mari. And for Zyzidium, I use cryopreservation, which means we store them in liquid nitrogen. So when you do cryopreservation, it becomes a lot more complicated because there's a lot of steps involved, and each of those steps needs to be optimized for each species. So we're looking at taking the embryos out of the seed. So you can see in the photograph at the bottom are 2 embryos in a seed. We need to excise that, grow them on in conditions that are sterile, remove the moisture, even though they are desiccation sensitive. We use cryoprotection for it, rapidly freezing, thawing, recovery, and regrowth. For the purpose of this talk, I cannot go into all the details for the cryopreservation, but the outcomes have been published in 4 papers, which I will summarize in 2 or 3 bullet points. The first one is that Zyzidium mari embryos are highly sensitive to desiccation. So when you start drying them, they rapidly die. When I go back to the DSC, which we used for Lophomertus, we can also see that swamp mari embryos need to be dried to moisture contents of below 0.25 gram per gram. to prevent the formation of ice crystals when we start freezing them. But this is lethal to the embryo, so they do not survive it. For this means we would need to look at faster cooling and warming rates and probably adding antioxidants to see if we can improve the embryos following desiccation and freezing, which is cryopreservation. So in conclusion, based on our DSC that we could see the lipid thermal fingerprints, conventional seed banking is probably not going to work for Lophomertis bullata, Obcordata, and Neomertis. We should therefore investigate storing these seeds outside of the lipid thermal transition temperatures, which is not at -20. So it might be in liquid nitrogen, or it might actually be higher temperatures than -10. For the cryopreservation of Syzygium miri embryos, it remains rather challenging because they are big and they really don't do well when you dry them. But the results used in the study can be used to optimize further cryopreservation protocols, and this includes the application of antioxidants to increase embryo survival and help the enzymatic antioxidant survival. Faster cooling and warming rates to prevent ice formation, combination of various methods in cryopreservation, which is rapid desiccation and protection through chemicals, and then optimizing cryopreservation through the embryo survival following encapsulation and dehydration. As always, there's a lot of acknowledgements, which I will just leave up there for you guys to go through. Thank you very much for listening.
Awesome, thanks for that presentation, Karin. We're now moving to the panel discussion, and joining us alongside the speakers that we've already had, Emma and Karin, is Albie Marsh and Taylor. Kia ora, everyone.
Kia ora.
So I'll just take this moment to remind viewers that we have the live Q&A function. So please drop your questions in there, and I'll just start working through the ones that have appeared already. This is a question from Susanna to Emma. Thanks for your interesting talk. Do you have enough genetic diversity amongst those 5 plants?
Yes, so we had the 5 individuals from the 3 discrete kind of populations. So we've probably got the best diversity we can, but we're certainly working on getting those other individuals into the ex situ conservation program.
And there's a couple more questions for you, Emma, actually. This one's from Monique. It's great to hear about the fantastic conservation mahi that you're doing. With the rata that got down to 12 individuals, is that figure only counting wild individuals, or is that including any that were captive in botanic gardens, etc.?
Yeah, good question. So yes, that is the total for in the wild. There are lots of individuals that are in cultivation throughout Aotearoa, in botanic gardens and home gardens and the like. But from the DNA work that has happened on those ones in cultivation, they actually are all genetically the same. So they've come from one individual. So using some of those for conservation is not as valuable as these 5 genotypes that we're currently holding and working on at Auckland.
And finally, Emma, this is from Stuart. Have you observed myrtle rust on M. bartoleti? Sorry for my pronunciation there. Are you returning plants grown from seeds and or all cuttings, i.e. is there greater diversity in the plants being repatriated? Yes,
so we haven't observed any myrtle rust in the Auckland Botanic Gardens, but I am aware that in other places that Metrosideros bartlettii or Rātamoehau has got myrtle rust, but I'm not sure on how susceptible they are at the moment. The plants that we're repatriating from Auckland, we grow those from cuttings, so we're trying to just preserve the genetics of those 5 individuals, and then Ngāti Kuri are carrying out those hand pollinations in the far north to build up the genetic diversity through seeds. So botanic gardens are quite limited in terms of space for growing kind of ex situ populations. So at least if we maintain the diversity from the wild, then back in the wild where we're repatriating, then those seed grown ones can be planted out there. I think that was everything.
And Karin, this is a question from Claire. Are there other New Zealand species which aren't able to survive -20? How long are you hoping to bank them for?
Yeah, thank you, Claire. I'm actually going to answer the second part of the question first and then the first one. So when you do seed storage, you are often looking at hundreds and probably thousands of years. That's what you're aiming to do. But whenever there is a need for that seed to come out to restore a species, obviously then you will deal with that. Now, and that should then mean when you store them, that must be the most optimal temperature for that very, very long period of time. So to answer your question, for other New Zealand species, there's actually not a lot of data available. It hasn't been researched well, but yes, there are some. So we already know which ones are similar to Maritawaki or Sazeja mari, and that is tawa. And there's also some concerns around kauri, which we are looking into a little bit more. And the concern for kauri is seed that's been sent to Q Millennium Seed Bank in Wakers in the 70s have been tested and they've lost a lot of viability already. So at Otari, we've got a collection of kauri and we are starting to look into it. And there does seem to be a couple of problems. So what we need is a lot of people starting to look into seed banking and see what works or what doesn't work for New Zealand species.
And Karin, on that note, I'm interested in the kind of logistics of this storing seeds. So you mentioned that you could potentially store them for up to 1,000 years. What if you needed to access them at different points in time in the future? Are they sort of partitioned into separate packages to withdraw?
Yeah, that's a good point. What we generally do is we've got a collection that's used for monitoring, and there's a collection that goes into long-term storage. Because as I said with kauri, and as we've seen with Rama and even Opkodata, so Rahutu, is you need to monitor your collections because we don't have the information. So they get stored in separate vials. Especially in cryopreservation, each vial will have 20 seeds, which is really convenient because then you can just withdraw them. Because what you don't want is taking your large collection out because you risk actually getting those temperatures up every time you withdraw something and then refreezing them or possibly reabsorbing moisture. So logistically, you need to work out your monitoring samples and your long-term storage samples.
And also speaking to the idea of storing seeds, I think it was on Tuesday in the Oranga theme, there was talk about seed banking and having a new kind of repository here in Aotearoa, and Te Tere Whakamātaki being leading that. I was just wondering if there were any connections going on with the work that you're doing at the moment, or see a future pathway there?
As future pathway, definitely, I think that is the only way that we can actually progress this effectively. There's some connections forming. I think the challenge once again is just there's so much information that still needs to happen, so much research that needs to happen. And it really makes me happy when I hear other people wants to do seed banking as well, and shows the importance of having good protocols that we all can rely on and make sure we can bank effectively. Especially when we get to species like Rama, who is starting to massively fail in reproduction due to myrtle rust. The race is on, and we need to find a way to secure them in seed banks to give us options for the future. It's not the silver bullet, but it is an option for us.
I can add to that, Simon, and that it's part of the theme 5 in this week program, or in this conservation restoration theme. So there are 2 different work streams operating in parallel. So the work that Karin and Jayanthi are doing, as well as the Oranga theme and their piece of work. Key to the work that Oranga are doing is working with mana whenua around the country to ensure the proper protocols are in place for seed collection, and then enabling those mana whenua groups that agree to that to have access to some of those seed should the time arise where some of their populations do become lost to them, I suppose. So, yep.
Awesome. And just looking at the clock, I think that might be all the time we have for our panel discussion. I'd like to send a mihi to Karin, Emma and Albie for joining us during this session. And I'll hand it over back to Taylor to take us through the rest of the session. Kia ora.
Kia ora, Simon. Yeah, and huge mihi to all the speakers again. A really, really interesting session. So now it's my honour to introduce Beccy Ganley. Beccy Ganley. Beccy is the Rākau Taketake science leader and a scientist at Planted Food Research. So kia ora, Beccy.
Kia ora, tēnā koutou katoa. Ka noho ahau ki raro I te maru o Ngāti. Ka noho ahau ki te taha o te awa o Utahina. Nō tawahi oku te puna. I whanau mai au I te papai I oea. E noho ana au I Rotorua. Ko Ngāti Pākehā toko iwi. Ko Beccy Ganley toko ingoa. He kaiara taki o ngā Rākau Taketake ahau. E mahi ana ahau ki te rangahau aha marakai ki te poki. So hello everybody, my name is Beccy Ganley and I'm the science leader of Ngā Rākau Taketake and I'm also a scientist at Planted Food Research. I've been working on myrtle rust in particular for a long time now. I started in 2010 when it was first found in Australia and have subsequently worked through the incursion and up to this very point. I've also been strongly involved in Kauri dieback over the years as well, from those early years where it was first found to be becoming a huge problem. Today I'm just going to talk a little bit about Ngā Rākau Taketake, where we've come from and where we're going to. And just wanted to also at this moment just take an opportunity to discuss why I'm talking today. I know I'm not part of the program that we had, but one of our speakers had to pull out because she was unwell. And I just wanted to really touch on these connections that we have between human health and plant health. And also I just wanted to just take a moment to reflect back on what we've had over the last couple of years with COVID. COVID has been a huge and defining, well, not a huge factor that's influenced our research. We'd started off and it happened in the second year that we were underway. It's influenced our ability to meet face-to-face. It's influenced our ability to have workshops and meetings and conferences. It's had a huge, huge role. But at the same time, it's really heartening to see that although it has influenced us, it hasn't defined us. And I'd like to say a massive thank you to all of the researchers out there and everybody who's been involved in Ngā Rākau Taketake. And that also includes hapu iwi, stakeholders, industry, end users, regulatory agencies, everyone who's linked in for what they've managed to achieve over this time when we've had all of these restrictions and quite a change from what we usually see. So there was just a very brief acknowledgement, and now I'm just going to continue on to what I was going to talk about today. So the first thing that I really wanted to go through was the common goal. So we're all a diverse group of people. We come from different walks of life. We have different values and different opinions. But the one thing that's really, really centric and what you see coming through really strongly with Ngā Rākau Taketake and those who work in this area is that we are put together by this common goal of saving our iconic trees, whether it's kauri, whether it's our myrtles. And that's really important, and I think it's been a really key part of how we've been able to come together, because despite our different values and our different views, we do have this one common goal. A key part of Ngā Rākau Taketake has been the inclusion of Māori from the very start. So all of our research is co-designed and co-led with Māori researchers, and you've seen our Māori co-leads through this week. We also work heavily with iwi, hapu and whānau, and you will have got a sense of that through this week of presentations. All of our themes have mataranga Māori embedded in the project, and we also have a huge focus of Ngā Rākau Taketake has been on building community trust. We've got our Koiora and Myrtle Ora community hui. If you're not part of those and you're interested in coming along, we'd welcome anyone through to these hui, which are a great way to share and connect across the wider community. We've also have use and dissemination of rangona and mataranga Māori guided by Māori. And our program is huge. We've got over 160 researchers from over 25 organisations. So what you've seen here over this week has just been a tiny snapshot. One of the things I wanted to touch on for a little bit was everybody plays a role. We're all individuals, we all have our roles, but together we come collectively, and that's what makes a difference. And this is a huge part of what Ngā Rākau Taketake stands for, is everybody connecting to work together. Part of this, as I mentioned before, are our community hui, and these have played a huge role in connecting and enabling some of the transfer of knowledge from research through to operations, and also back from operations through into research. It will be a huge part of NRT's legacy, these community huis, in a different way of how scientists can work with a wider community. We also just wanted to mention that we have some of New Zealand's best scientists who work in our program, and what's been fantastic is how we've partnered up some of the science expertise with our new generation of scientists that are coming through, and that experience for these new generation of scientists to work with those who've experienced and have a long history in the science sector has been invaluable. And I think it's really, really important that we see that part of what we've got coming through for that next generation, whether it's work on myrtle rust and Kauri dieback or whether it's on a different plant disease. There's been a huge upscaling of capability and capacity across New Zealand. Our research being taken up by next users is a critically important part. We've already seen a lot of our research that has been taken up. Sometimes it's very applied. Sometimes the next user is another scientist who'll be taking it on to the next step. But it's a really key part. Although we're coming to the end, in particular for myrtle rust, and we're not sure what the future will hold, the funding for myrtle rust ends in June 2022. We're hopeful that we're going to see further work in this space. We've started to make some really, really key progress in how to manage and control this disease, but the fight isn't over yet. Myrtle rust is a serious disease, and it will be a national priority for a long time. So if any of you happen to be in a role of influence, be more than happy for you to bang the drums for further research and also operations, and that's been part of what's really been missing from the whole of the work program that we have. It's fantastic we've got Ngā Rākau Taketake sitting there as our science arm, but we really need to have a strong operations arm next to it, and it's that coordination of the two that work together to have some really, really key outputs. But having said that, we have amazing community of end users, hapu, iwi, stakeholders, industry, agencies who do uptake this research, and that's really, really fantastic to see. And I think that will be another part of the legacy, is watching these research programs and the research that we've developed to be taken up. I also did want to reflect on the new tools that have led to new opportunities and will lead to new opportunities to control or manage disease. Over the week, we've heard many of these new tools being discussed, and one of the ones in particular I wanted to mention was RNAi, or RNA interference. This is something that we haven't had time within Ngā Rākau Taketake at this point to actually see this through to implementation as in an applied tool, but we've started down that pathway, and it's fantastic to look back on the rich history that goes behind it. It starts off in some of the earlier programs where it was the genomic work on Osteopuccinia ciliae, the pathogen that causes myrtle rust, and we saw that this tool, the early steps which would potentially lead to a tool like this being developed and potentially implemented. And watching that research come down this pipeline and seeing where we've got it to today is fantastic. As I said, we're not at the point where it's at a level for implementation, but we've made some really, really key steps. And some of that key steps has been connecting through to hapu iwi, looking at the social license, looking at what issues might there be if we were going to implement something like this. And we can see that pathway forward of where we can take a tool like that. And it's not just for myrtle rust or Kauri dieback, but it also has applications to other diseases that we have in New Zealand and could be taken by other industries and used for different purposes. But it's the whole science that goes around it and how we can progress along that. RNAi for New Zealand would be a new to New Zealand tool. It is used overseas, but it's never been applied in New Zealand. We've had an EPA ruling which has said it's not a GMO, which has opened the door for it to be considered as a control tool. So we've got some really, really promising opportunities out there, but there is still more work to be done, and looking forward to seeing where we get to in the future with these projects. The final thing that I wanted to touch on was te whakahongona, and as I've put here, this is a new way to undertake science in Aotearoa, New Zealand. I know that there was a session previously, and hopefully you've all had a chance to listen in to hear what's been happening in te whakahongona, but it really, really is heartening to see some of these new steps. It's a completely different way of how scientists can engage with hapu iwi and make a difference. Yes, this week we met with 45 mana whenua kaitiaki from across New Zealand, who we have taken through to make some of the first steps in seeing the reality of te whakahongona in action. And that involved them meeting with the scientists. We're discussing the disease problems, discussing the research, discussing their role in this research program. Behind all of this are the cultural authority agreements, that cultural license, which means that there's that respect of the scientists going into the rohi for hapu iwi. Someone yesterday asked me what success would look like for te whakahongona, and I'd never been asked that before, and I had to think for a little bit, and my response was that it changes how we do science in New Zealand, and I think it's making a difference already. Even if it's not te whakahongona, some of the ideas and the practices from this is starting to spill out into other research programs. And what I would love to see, and what I would see as a success, is if we have not necessarily te whakahongona in itself, but the principles behind it just sit behind everything that we do in science, and it changes how we're doing things, and it makes a difference. And not just for New Zealand, that it stretches out beyond that, and it has an international influence for other Indigenous people. So on that note, I'd just like to wrap up. Thank you all for coming along and listening to what we're doing in Ngā Rākau Taketake. What you've seen this week has only been a tiny snapshot of what we do within Ngā Rākau Taketake. There's a lot more that goes on, but we just couldn't present it today. So I have put up here the link to our newsletter. We regularly put out the highlights and new information, what's happening across Ngā Rākau Taketake. So if you're interested in finding out where we're at, what we're doing, please, I'd encourage you to look at the newsletter. But I'd like to thank you all for coming. Ngā mihi.
Kia ora, Beccy. What an inspiring kōrero. And yeah, I'll definitely be signing up to that newsletter to check out what's happening. We don't have time for questions live now, but if you do have any questions for Beccy, you can chuck them in the Q&A and she can come back to them and type out some answers to you. We're now just moving on to our final speakers of the day and of the conference. And yeah, they shouldn't need introduction as the co-directors, but I'll give one anyway. So we've got Daniel Patrick and Shaun Ogilvie, sorry. Daniel joined the team in December of 2020, and from 2011, he was the executive director of Ngā Pai o Te Maramatanga, New Zealand's Māori Research Excellence Centre, hosted at the University of Auckland. And we've got Shaun, who is of Te Arawa and Ngāti Awa descent, and is the Biological Heritage Kaihautu Ngātahi, the co-director Māori. Daniel, sorry. Kind of a bit scrambled there. Shaun works closely with co-director Daniel Patrick on strategic priorities and building strategic links with Māori and Indigenous researchers, groups and entities. In addition, Shaun is the Māori Business Development Consultant for Cawthorne Institute, a principal investigator at Ngā Pai o Te Maramatanga, and a contractor to other organisations including Lincoln University and the Environmental Protection Authority. So kia ora to both these speakers, and I'm really looking forward to hearing what they have to say.
Kia ora koutou katoa, he tino mihi ki a koutou anō. Just adding to that too, thank you for that, Taylor. Just adding to, I'm also a professor at the University of Canterbury and at the Ngāi Research Centre. I better add that in because my boss is at the Ngāi Research Centre, might not be impressed. So kia ora nō koutou katoa. Yeah, just really quickly, as already introduced, Daniel and I are the co-directors of the Biological Heritage National Science Challenge and Ngā Rākau Taketake. And you will have learnt from this week that it's really is a large program of research, and it's a pleasure for both of us to be able to lead it. And I know, I mean, I speak for myself, but I'm sure I speak for Daniel too, that it's actually personally rewarding to be able to make our own contributions to the mission of these programs, and that's to reverse the decline of New Zealand's biological heritage. So while there's a lot that we could actually talk about today, we decided to focus on one and particularly important area, and that's around the power of collaboration. So we go to the next slide there, Daniel. So big picture stuff in terms of collaboration, we really are talking about a massive collaboration exercise here. There's more than 100 organisations involved in the work of the National Science Challenge and Ngā Rākau Taketake. We've kind of administered actually more than 100 research projects over that time, and within the organisations there's many iwi, hapu and Māori entities, and we've supported more than 250 researchers over that time too, and it's global. So just as an example, we have an international science advisory panel, and so we've got support from around the globe really. I'll move on to the next one. So we just wanted to focus in on 5 vital areas that has kind of come to light for us as being really important in terms of achieving our mission. They're all underpinned by collaboration, and those 5 areas are legal and policy, we'll call it that, a maturing of the relationship between mātauranga and science, having real conversations about new technology and emerging technologies that are coming through within the challenge. That transition from research and new discovery to implementation, so the adoption of new research and discovery to be utilised beyond the kind of research phase, and then of course future generations. So I'm going to talk a little bit more about the first 2 vital areas here, and then I'll hand over to my fellow co-director to talk about the other 2. So legal and policy, I was earlier in the week, back on Tuesday, I got to admit I was pretty heartened to listen to Arawa Mead's presentation, and her photo that she put up there of her meeting Nelson Mandela, and the quote there from Nelson Mandela: "I see no future for parks unless they address the needs of communities by taking communities as equal partners." And so the conceptualization of partnership and te tiriti, really, really important. So in addition to the words that Arawa Mead shared with us on Tuesday, and also again this morning from Maria Barge and Carwyn Jones, there's a real need for an overhaul about the way we actually are committed to te tiriti. And to steal some words from earlier today, we need to retire old colonial thinking and restore respectful, balanced relationships. And that kind of neatly segues into the next of our vital areas. So if you can do the next slide there, Daniel. And that really is around maturing the relationship between mātauranga and science. So for me, this is an area that I've been working in for many, many years. And I think there's a real need to be kind of dropping the us and them kind of way of thinking. Jamie Ataria talked earlier in the week about there being a recent push from some members of the science community who felt the need to act, and I'll quote them I'll quote the title of a recent letter to the listener. They feel the need to act in defence of science, and so the whole way of thinking about having to defend science from mātauranga is really, to me, it's just a false way of thinking. It's a kind of us and them thinking, and it's a mentality that puts mātauranga and science in direct competition. And as I said, I just think it's simply false. For me, and I guess there'll be different opinions on this, but for me, I see science as a tool. It needs to be thought of in the sense of how can science help kaitiaki to do their job better? How can science support us as kaitiaki to do their job better. So I'd just like to talk about a concept that was raised by Hami Piripi as well, and that's moving that kind of relationship to a more matured place. And he used this phrasing. He talked about being happy versus being contented. So in a happy relationship, you're kind of putting a lot of work in to make sure that both partners are happy in the relationship. But in a contented relationship, it's kind of matured enough so that it kind of gets to a point where there's a mutual understanding about what each partner brings to the relationship, and so a contented trust that each will be there to support the other. So I think we kind of need to be thinking along those lines to get to this point where science and mātauranga can kind of work together in a contented, tiriti-based way. So kia ora koutou, I'll hand over to my fellow co-director.
Ngā mihi, Shaun. Tēnā koutou katoa. So another one of those areas that have emerged through, I suppose, today and through our observations, and together Shaun and I have discussed the real need for conversations about new technology. Within the gene tech space, there are emerging technologies that are really exciting, and we push your attention to a particularly pertinent example, and that was shared earlier in this conference, the double-stranded RNA gene silencing tool that has shown real promise to reduce the reproduction rate of varroamite, which is a destructive pest for our honeybees. And as Tama Blackburn, kia ora Tama, stated earlier in the week, the way forward is for communities and researchers to have real conversations about these technologies and to effectively co-develop them from the outset. It's hugely important that we as the challenge bring these conversations and discussions together as an independent and neutral voice. And that's something that we are challenged to do and will do. So the fourth vital area we raise, transition to implementation. And this is a serious problem for our research outcomes. We need them to be implemented or operationalized, operationalization of our research. And as Desi Ramu terms it, avoiding the valley of death for research. We want to avoid that. And the time that we have remaining in the challenge, we'll be working hard to ensure that the research outcomes are adopted and owned by communities, agencies, businesses, regional councils, and other organisations for the ongoing benefit and impact, the impact to our biodiversity, to our biological heritage. The fifth area, future generations. This is the final vital area. Focus on future generations, and we're immensely proud of Ngā Pikarere, and there's been a number of sessions there and acknowledgements of the early career network of biological heritage that I hope spans further than that, and others are welcomed. And whether you noticed or not, we've made a conscious effort during this week to have our early careers chairing the sessions and presenting some of their terrific mahi and insights. It's been wonderful to hear them. Get their fresh ideas, get their insights on the research that they're undertaking for the challenge and across, I suppose, the community. They offer different perspectives and high level of energy. Also, capability and capacity building, rather. Capacity building has to be part of our thinking. This is especially true for building Māori capacity, and there's an ever-increasing demand from a wider pool of organisations for this capacity, for Māori capacity. So just in closing, as we move toward Matariki Rising, while we've got a public holiday to reflect that on the 24th of June, it's a time to be thinking about renewal and crazy and ambitious ideas. We need to think, take time to think and reflect. And over the past 4 days, reflecting on that, we've seen a sample of the mahi of our many hundreds of researchers, community, iwi, hapu, and organisational partners. Our tiriti-led approach is evident throughout, and we want to thank the presenters and all of the others involved for their commitment to reversing the decline of New Zealand's biological heritage. The mission is large, and much remains to be done, but collectively, with the power of collaboration, we will achieve our mission. Really, we must collaborate. These vital areas, these headline items won't be achieved without the power of collaboration and working together. And as Beccy just noted, everyone plays a role. Everyone plays an important role. So as we bring Crazy and Ambitious 3 and the Kauri Land Summit for this year to a close, we've got to express a huge thanks to the many speakers, the communities, researchers involved, the keynotes and guest speakers. Their sharing has been fantastic. You, the delegates, for your participation, your engagement, and importantly also the team behind the scenes, those fantastic videos for Kauri Land, and those in our support team of biological heritage, supporting Ngā Rākau Taketake to bring this conference and online event together. And in particular, a shout out and acknowledgement of Stacey Bryan, our science communicator, for really creating, leading and managing this event. Ngā mihi nui kia koutou katoa. Thank you. Next year we look forward to, or in future we look forward to meeting in person. Next year we will have our Kauri Land Summit, in 2023, and Crazy and Ambitious 4 will be in 2024. We trust that the circumstances will allow us to meet again in person. But in the meantime, let's connect and collaborate as we can to support our collective genius and help our biological heritage to thrive. Mauri ora kia koutou katoa.
Kia ora kōrua, Shaun and Daniel. We just have one pārtai from Denise. When does the National Science Challenge finish, particularly for Kauri dieback?
Oh, kia ora, Denise. I'll answer that, and Beccy would probably be a good one as well. But Ngā Rākau Taketake actually finished in terms of the funding June 2023. We have got, due to the COVID impacts, a slight extension on that, but no new funding. So work will continue through that period. Beccy, any?
No, but would welcome any support for new funding for research, but also for anything in the operations space or anything that will just contribute towards seeing outcomes that we want to see for Kauri and Myrtles.
And I think that's hugely important, and Beccy touched on that, the significance of the strategic research, what we've heard to date, just from this hui, this event itself. There needs to be a future, there needs to be a pathway, and we're working closely and making all attempts to operationalise, work with Tia Kina Kauri. Beccy and the team are doing some fantastic work, and we hope that it's extended and there is the operationalisation or implementation of those research outputs.
So that when the challenge ends, that there's still that legacy that some of the work can continue.
Absolutely.
Oh, well, fantastic. Since there's no other questions in our Q&A, I'd like to again thank all of the speakers across the conference. It's been a really amazing experience, and it's been fantastic to hear all of the mahi that's been done across Aotearoa. I know I always leave conferences like this with a real sense of optimism and excitement, just knowing there's all these awesome people across the country and all this awesome mahi being done to protect our species and our landscapes and our culture. So thank you also to the team who put the conference together, and thank you all for tuning in. I'll hand over to Shaun to close us with the karakia.
Kia ora no koutou katoa. Ko taku pirangi ki te mihi ki a kōrua, ko Taylor, kōrua ko Simon, mō tō mahi rangatira i te ahi nei no reira. He tino mihi ki a kōrua. Ko te karakia whakamutunga, kia tau, kia tātou katoa, te atawhai o tātou ariki, te aroha o te atua, me te whiwhi ngātahitanga ki te wairua tapu. Ake. Āmene.
Kia ora. Ngā mihi.